Hello, welcome. This is the iGEM BioBytes podcast. I'm Aarush, and
I'm Jamie, his co-host.
today's episode is going to be all about spliceosomes. Jamie, do you know what spliceosomes are?
Well, I've done a little bit of research, but I was hoping you could enlighten me today.
Good. Well, in that case, can you tell me how much you know so far?
Okay, so I know spliceosomes are large ribonuclear protein complexes that are found in the nuclei of eukaryotes. Do you mind quickly just telling me what a ribonuclear protein actually is?
Cool. So, a ribonuclear protein is essentially any complex made of both RNA and also protein that's found in the nuclei, obviously, of eukaryotic organisms. They can have a wide variety of roles. One of the most famous ones is obviously the ribosome, which completes translation of mRNA into polypeptides.
So, what does the spliceosome actually do?
Based off the name, what do you think it does?
Well, I know that splice means to cut out certain things. So maybe it has something to do with cutting out pieces of DNA.
That's sort of correct. So essentially, what the main role of the spliceosome is to cut out the introns out of pre-mRNA so that it can be made into mRNA and then translated into a protein.
What's pre-mRNA?
Well, what we're taught in our biology lessons is that the gene is transcribed by an RNA polymerase into mRNA. But in reality, what actually happens is the gene is transcribed by RNA polymerase into pre-mRNA. And this mRNA actually contains all of the introns which need to be spliced out so that then the protein, which doesn't need these introns, can then be expressed by the ribosome.
So what's the point of removing introns then?
Eukaryotic genes have both introns and extrons. The introns are the intervening sequences, which don't tend to have a role in the eventual protein. But, however, a process called alternative splicing can occur. Have you found out anything about alternative splicing in your research?
I have actually. I know that alternative splicing allows one gene to produce multiple protein isoforms, and that increases proteomic diversity.
Hey, could you explain what proteomic diversity and isoforms are?
Oh yeah, sure. So isoforms are slightly different versions of a protein that are produced from the same gene, but they differ in some kind of structure or function. And proteomic diversity is the variety of isoforms produced by a single gene.
Well, what function does this serve in a cell, for example?
Well, proteomic diversity is pretty crucial because it allows organisms to produce this wide range of proteins with distinct functions. And that allows for adaptability and complexity and specialization in biological processes. A great example of the importance of proteomic diversity is the human tropomyosin gene, TPM1, which produces multiple protein isoforms through alternative splicing. Tropomyosin is a structural protein that stabilizes actin filaments, and its isoforms are tailored for specific tissues.
So this is specifically for muscles?
Yeah. So in skeletal muscle, one isoform supports muscle contraction, and in smooth muscle, another isoform regulates actin dynamics for different contraction patterns. And then in non-muscle cells, isoforms help maintain cell shape and intracellular transport. This diversity ensures that the same gene can meet the unique functional demands of different tissues. However, dysregulation of tropomyosin splicing can lead to diseases like cardiomyopathies or cancer, where the wrong isoform is expressed in the wrong tissue at the wrong time.
That's so cool. Would you like to learn a fun fact?
Yeah, sure.
Humans have 20,000 genes, and yet we can make over 100,000 different types of proteins because of alternative splicing.
Oh wow, that's a lot of isoforms. I guess spliceosomes are a really crucial part of cell function.
You talked about dysregulation a few moments ago. How can dysregulation lead to disease?
Sure. So one way is through the missplicing of critical genes. So for example, mutations in the spliceosome components can cause improper slicing, such as intron retention or exon skipping or activation of the incorrect splice sites, and that leads to defective proteins or altered protein isoforms.
That's so cool. For example, in the condition microcephalic osteodysplastic primordial dwarfism type 1, the incorrect splicing of a specific gene causes a much smaller body, but specifically affecting the head, which can have terrible effects for brain function and the growth of many different organs over the course of the person's life.
Oh, that's so cool. There are also other types of dysregulation, like genomic instability, where mutations can activate DNA damage pathways and impair the cell cycle progression, which leads to apoptosis. There's also mutations which disrupt splicing of genes that are essential for specific tissues, like tissues in the retina or in myelodysplastic syndromes. And then there's also immune dysregulation, where the incorrect splicing of immune-related genes can lead to dysfunction due to bad isoforms of proteins, such as the one that activates B and T cells and differentiates them and allows for cell signaling. So, Aarush, how do spliceosomes actually work?
There are a few steps that the spliceosome goes through to be able to splice the introns out of the pre-mRNA, and I'll go through them now. Firstly, two specific ribonucleoproteins called U1 and U2 land on the pre-mRNA, and they hybridize with the specific nucleic acid sequences at the start and the end of an intron.
Okay, what did all those fancy words actually mean?
Okay, so firstly, hybridize. Hybridize essentially means come together with a complementary strand of DNA and temporarily sort of bond to it. An intron is an intervening sequence, as we've discussed before, and at their beginnings and their ends, they have specific sequences which essentially signal to the spliceosome where it can bind and where the intron can be spliced out. Then, over 7 different steps, teams of ribonucleoproteins join and exit the spliceosome, along with a vast array of protein partners. In particular, a troop of protein helicases wind and unwind the mRNA to arrange all of the base pairs of the ribonucleoproteins and completely change their conformation. What these helicases do, they essentially become the driving force of the various remodeling steps of the spliceosome, which helps the spliceosome to not only be specific, but also very accurately do its job of splicing out the intron.
So how does the splicing chemically occur?
While the helicases are doing all of these rearrangements, this pulls the mRNA's 5' splice site close to a very important adenosine base called the branch point site that is located inside of an intron. At the hydroxyl group on that adenosine base, the hydroxyl launches an attack on the phosphate group at the first intron-extron border. This severs the intron from the extron via a transesterification reaction. After another massive spliceosome rearrangement, a second transesterification reaction occurs, so the intron is completely severed from the gene and excised. Eventually, the remaining exons are ligated together by the spliceosome, and the removed intron becomes what's known as a lariat, which looks a bit like a lasso.
Wait, I'm curious. Do you know how the spliceosome was discovered?
Yeah, essentially, in the 1970s, after Crick and Watson has discovered the structure of DNA, there was a lot of discussion around how it might work in eukaryotes, because of all of the experiments that had actually been done in prokaryotes, which obviously don't have the spliceosome. What happened is, when studying eukaryotic cells, such as yeast, the molecular biologists discovered these lariats as round pieces of RNA and were really confused as to where they came from. Then, in 1977, some biochemists looked at the length of the mRNAs that came from DNA genes in eukaryotes and found the mRNAs were suspiciously shorter than the genes they'd been transcribed from. A lot of experimentation was done, and eventually the spliceosome was discovered, and 2 major biochemists discovered the Nobel Prize for it. Well, I've talked about the history of spliceosome research. What's the current research like?
Well, as of October 2024, researchers have created the first blueprint of the human spliceosome, which is pretty crazy because it's a really complex and intricate molecular machine. It took more than a decade to complete, and the research reveals many new drug targets that could benefit a wide spectrum of diseases, including most types of cancer, neurodegenerative conditions, and genetic disorders. How do scientists think spliceosomes evolved into what they are today?
There are 2 main competing theories. One of them takes into account what's known as the RNA world hypothesis. What this hypothesis hypothesizes is that all life on Earth initially didn't use any sort of DNA and also didn't use any sort of protein, but initially completely relied on RNA. including for enzymes. And the spliceosome is actually a type of enzyme called a ribozyme, which is made of RNA, because the introns in a spliceosome actually catalyze their own splicing, which is a very interesting piece of history, or so some people think. Other people think introns develop later, because obviously they are only found in eukaryotic cells, which means there's a good chance that eukaryotes evolved them by themselves in order to increase their proteomic range without increasing the number of genes. Many evolutionary biologists also think that the spliceosome itself evolved from bacteria's self-splicing introns, because some bacterial species actually do have introns, but these introns remove themselves once the mRNA has been transcribed, and move around the entire genome, splicing themselves into different mRNAs as they go, which many evolutionary biologists think is what the spliceosome structure actually evolved from, as it also has many different individual elements which come together to form the entire spliceosome.
Okay, so that brings us to the end of our discussion on spliceosomes, the molecular machines that play a really crucial role in RNA splicing. We've explored how they recognize and remove introns, the intricate coordination of their components, and the impact on gene expression in human health. As research continues, scientists are discovering new details about their function, potential links to diseases, and even ways to target them for therapeutic applications.
Thank you so much for listening to the iGEM BioBytes podcast, and I hope you've learned something new today. Stay tuned for more episodes.